Membrane.
Membranes. Livers are a type of
Membrane.
membranes. Okay? Physiology is differences in membranes. That's it. So when you start learning about how things actually work in tissues, these are all about the structural levels of the membrane. So we have a membrane, and inside you'll have a nucleus, right? We'll have our chromosomes. And what else do we have? What other structures? Rough ER, right?
Rough endoplasmic reticulum.
Smooth ER,
Mitochondria.
Golgi,
Mitochondria.
mitochondria, right? Lysosomes,
Did we say Golgi?
ribosomes, you have all those other functions, right? And so, if you're going to communicate with something, you're going to release a signal.
Mm-hmm.
Okay, what are some signals that we've talked about so far?
Noggin and BMP4.
Noggin and BMP4. Okay, how do they communicate with another cell? A receptor, right? So they have a receptor on them. Or if they have an antibody on them, they have something on them that they can receive information. Okay, that is a lignin bound or it's a protein. So we have a protein, then we'll release a receptor, right? And so that is one way to communicate. As we are going to dive in today and do next week, we're going to think about how do action potentials work. How do changes in membrane polarity work? If I change the electrochemical charge of a membrane, does that change its shape? Yes. Can we do work with the change of shape? Yes. All organs are, all action potentials are, all any of this is a change of membranes. So if you think about it in that context, it becomes way easier than just memorizing what each one does and how it does it. Now you're going to need to know all that information. But if you contextualize it as all it is a change of membranes, it'll make a lot more sense. Okay, so membranes have channels. They have pores. Cells have gaps in them. There's gap junctions. There's all of these things that allow signals to pass through. Facilitated diffusion. Okay, we're going to go from high to low concentration. We're going to go through with a certain shape, certain size, certain charge. We're not going to let other things leave. We've talked a lot about these protein channels. We know how their shapes or sizes works, how the primary structure dictates that. And so we have a lot of those. There's going to be receptors on the inside of a cell in the membranes or on the outside. Noggin and BMP4 was on the outside. Do we have any examples of things in the inside? Tom, sure. Do we have any signals or anything that we talked about? Do we have any other signals? There are hormones. We haven't done a lot of hormones yet. Where's some other things? Let's go back to the lac operon. Tell me what's the lac operon again? Gene expression controlled by
Repressors.
lactose.
Lactose.
So, do we think lactose could be a signal in this case? Yeah. Right? So things that go through inside that's going to bind to the operon. Okay, so arrival of a signal. So you can have lipid soluble or lipid insoluble. We can have things that go through channels or things that bind to receptors. So in this case, lipid soluble. Let's do a steroid. Steroids like cholesterol, testosterone, estradiols, those were all soluble because they were mostly these long carbohydrate chains. They're going to pass through the membrane. They're going to bind to a signal receptor, often what's called a secondary signal. And it's going to transduce it into the target. Okay? So we saw examples of like this would be very similar to the lactose example. We have another type where it's lipid insoluble. It's going to bind to a receptor, which is going to trigger a cascade, which is going to release secondary signal molecules. And that's going to bind to a specific sequence. This example would be something like the TGFB with noggin and BMP4. Okay? So we have a signal receptor on the outside. It changes shape. It's going to do something. It's going to trigger a secondary response, which is going to make it become neurulation or skin.
Does the G protein steps, does that also activate a phosphorylation cascade, or is it just the enzyme?
Man, why'd you make me skip steps? So G protein. We'll go back and do our questions here in a second. You blame Carrie. So G protein is inactive until a signal is received. At that point, we're going to do a phosphorylation. When we phosphorylate something, we can activate or inactivate it. Okay? That's going to cause a secondary response because we're going to phosphorylate. We're going to work on a substrate. This is an enzyme reaction. So instead of just releasing a cascade, this is an enzyme. We're going to catalyze reaction, which then is going to trigger a response, which is going to activate something. So here is a chemical cascade. We're going to phosphorylate. We're going to change the shape of something else. We're going to catalyze the reaction and binding of two things together. Those two things allow the reaction to take place. An example of this, again, was in the lac operon. This would be the gas pedal. When ATP is present, we are phosphorylating. We are not allowing the reaction to go fast. When we take away ATP, the catalyzation is going to go quicker. It's going to bind to more of a higher affinity, and the reaction is going to go forward. These are kinases. Okay? Any questions on that? All right, let's go back around. All right. What's the correct one? About 10 seconds. All right,
Reception, transduction, response.
Reception, transduction, response. I picked that.
So we are going to receive, we're going to transduce the signal, and we're going to get a
Response.
next question. All right, talk to your chair.
Target specificity is determined at the level of a receptor molecule.
Yeah, that's what I put.
Target specificity is determined at the level of the receptor made on receptor molecule on the surface of the cell.
All right, really proud of you all. We got 100% on that one. Yeah, we did. It was one of our actual true first 100%. I'll show it to you guys so you believe me it was 100%. Now it would be funny if somebody changed the register.
I was about to do that.
The amount of smart assery does increase as you enter this. Legitimately, people are like, yeah, I'll sacrifice that point. It's as we continue to look at the signals, not all signals just do one thing. Okay? So, for instance, if you get scared in the woods, your adrenaline will spike. Okay? Adrenaline is a great example where it'll do one thing on one tissue, but something completely opposite on the other. Okay? So, for instance, I'm going to increase blood flow, if you have adrenaline, to my muscles. I'm going to decrease the blood flow to my gastric system. Why? So talk to her. Why increase the blood to my muscles but decrease it to my gastric system?
So you could run away, push something. So you,
Gastric system doesn't really do anything for running.
yeah, or pushing.
So
Or kicking
adrenaline is
around.
going to do two things. We're going to, well, it does many things. But in this example, we're going to do two things. We're going to increase blood flow to our muscles. Why? I need to run away. Okay? I'm going to decrease to my gastric system. Why? Because I don't need to eat. I'm going to die if I don't run away. The food in my belly does not matter. So I can move that energy. I can move that blood away from it to provide the need at the moment. So this is a great example. So a signal is received, okay? Signal A is going to inhibit B, but stimulate C and A. Okay? So that would be a great example of adrenaline, right? And so what it could be is that the receptors do different things. It all receives the same receptor, but the response is different. But you could also have where C does nothing besides stimulate B. So if you have C1 activated, it'll stimulate that. And so you can get different cascading events. Okay, so there's lots of different components to this. So another great example of this is oxytocin. Okay, oxytocin is another hormone molecule that we're going to cover. Oxytocin is what makes you feel good. It's the feel-good molecule. But it will also trigger things like lactation. It triggers muscle contractions in delivery. So not having a high level of oxytocin means not only are you not going to bond with the child, but you're not going to be able to deliver the child. So it's probably cesarean, right? So we got to think about how all of these play a part, and it's based off of different receptors present. Answer that one. You answer this one because we already have 24 people. So, and then we'll find this question.
I didn't realize why. I was like, HDL?
Okay. Closing up this one, we'll talk about it and then re-answer this one. All right. They bind to the inside. Okay. So lipid insoluble bind to the outside. Now we'll go to this question. Okay. Couple more seconds. All right, talk to your.
Peptide receptor mediated endocytosis. If you're looking through all of them.
Yeah, it's like you show for their business, right? And you see something nearby, it's kind of like.
It is. Okay, doctor.
A. What?
The substrate.
Yeah, it looks like it from the diagram. B is the thing that grabs the substrate, essentially.
Receptor?
What does A and B represent? Reception. C represents transduction. D represents response. Which one is the signal? A is the signal, therefore signal receptor is B. The correct answer is B. I give you guys a really hard question, you get 100%. I give you a really easy question, we're 50%. That's how we roll around.
Maybe you train us for the hardest scenarios.
So, what we're going to do is we're going to understand how voltage, and voltage is measured on the membrane. So we're not talking about like actual like volts, but like the difference between the two sides of the membranes. So the potential of that. How the change of ions will cause the membrane's potential to change. And when the membrane's potential changes, it will change the shape of proteins. Okay? And when we typically mean change the shape of proteins, it means it's going to do things like open up a gate. And when it opens up a gate, more ions will move in, or ions will move out. Something will change. And so we'll have a relative difference between the two sides of the membranes. And only certain ions can go through certain channels. So, for instance, if we have a potassium ion, which is the charge there?
+1.
+1. It's going to move through an ionic channel that's going to be coded in
Negative.
negatives.
Negative.
Excellent. So it's going to move from high concentration to low concentration. That's a channel. So that's going to be facilitated diffusion.
Facilitated
Then
diffusion.
we're going to have pumps. Pumps are going to be active transport. They're going to move specific ions against concentration gradients. Okay? All of this will take part on the membranes. So, for instance, if we have the voltage is positive, okay, this channel is going to be closed. Sodium will not be able to go across. But if I change the membrane's polarity, it will allow that gate to open and sodium will
Enter.
enter. It'll go through. Okay? And that will change the polarity even further. That will change the voltage. So, the best way to describe this is imagine you have a big cardboard tube or a pipe. And you just drop a marble in at one side. Does it come out on the other? Probably not, right? But if I fill that whole thing completely full of marbles and put one in on one side, what happens on the other side? One pops out instantaneously. Okay? And so that allows communication to go very quickly. Because if you just imagine a really long cell, what's the biggest cell in your body in size?
Skin?
Egg.
An egg in volume. What about in length or in actual size, the surface area?
Neuron.
A neuron. You have a neuron from your head to your pinky toe. Okay? Probably about 2 meters in length. If I were to allow diffusion to take place, and I just have a signal get released, how long will it take for it to get down there? Have we had physics yet? No. This is actually one of the few things we've learned in physics. This is called Reynolds number. Okay, Reynolds number is a diffusion constant. Things are really slow. It would take years for it to get there. Is that a good way to get a response? No. But if I do this in a way with the voltage, I can get instantaneous response. And so the change of these gates will really allow for really instantaneous changes. All right, talk to you.
I put sodium.
Sodium. From these diagrams.
It was either sodium or no, potassium.
Because there was that. One of the questions in the quiz.
Okay, what from this illustration to this one, what's the difference? So this one and this one. Can sodium just go through?
No.
No, it is gated. Can potassium just go through?
Yeah.
Yes. So which one's able to go through easier?
Potassium.
Potassium.
Okay.
Okay, we'll do that question. Actually.
Oh.
Ooh, we're doing super good. This is exciting. We're doing all right.
Where's the sleep?
Which one is the most against? Sodium, potassium. Okay. This is going to go from high to low concentration, high to low, high to low. This is going to push it against its concentration gradient, so it's going to move it upstream. So it's going to require energy. So potassium is just flowing. It's going to want to go to equilibrium. But the sodium, as soon as it changes in polarity, we go to negative, what happens to this gate? Opens and it floods in. Okay, so that's going to be a change in the membrane potential. All right. So you've often seen the right? That's an action potential. Action potentials are governed by a threshold. All right. How many of you are wearing underwear right now? You knew that you're wearing underwear? Now all of a sudden you're thinking you're feeling your underwear, right?
You already did this.
Okay. What happened? I know I already did this. What happened? That threshold got lowered. If you're ignoring it, the threshold gets
Higher.
higher. Okay? So responses can be varied. So in this case, you feel something, a thorn, right? All of a sudden, you get a response. You've reached that threshold. We're going to depolarize. We're going to depolarize and go to the positive. Okay? More positive than you were already at. It's not, we're moving positive, we're just more positive. Then we're going to do repolarization. Repolarization is we're going to take it back to the original. But the problem is we're going to overshoot. This is called hyperpolarization. Part of the reasons why we overshoot is our pumps are really good. And they're going to pump so fast that it's going to overshoot. It's going to take too much away from the other side. And then we're going to get back to our resting phase, which then we can do it again and do it again. And each one of these occurs in a certain amount of time. All right? An action potential is either generated or it is not generated. Purely. That is it. We either generate it or we don't. What changes is the amount of them and the frequency of them. That is it. Action potentials either just generate or they don't. That's it. So, for instance, if you close your eyes and you're in a dark room, why can you see little flashes of light occasionally?
A light was turned on. Either that or.
Close your eyes. Every once in a while you see a little flash of light.
Oh.
Why? All reactions are spontaneous. Every once in a while, a action potential will generate. You're sitting there and every once in a while you get a little twitch. Why?
Action
All
potential?
reactions are spontaneous. Things will just happen. A neuron will fire occasionally. That just happens. It's going to be rare. And will you get a huge muscle contraction? No, because maybe a single fiber fired. You get a single cone depolarized. Or, in eyes often, like a subatomic particle might hit your eye. For instance, a little X-ray or something, and that will trigger. It's not in the visible spectrum, but it'll still trigger. So they're all or nothings. It will either happen or it won't.
Concentrate K plus concentration gradient favors K plus diffusion, while the electrical gradient.
Yeah, well, that's what I do.
When a neuron is at rest, the potassium concentration gradient favors potassium diffusion out of the cell, while the cell electrical gradient favors potassium diffusion into the cell. A is the correct answer. We have something is occurring. Okay, these are called failed. It didn't generate enough of a threshold to actually move forward, so it just nothing happened. We get a depolarization, so stimulus reaches that threshold. We get depolarization. Potassium ions flow in. That membrane has changed polarity. Okay, that ion gate has opened. Those ions flow in. That consequence allows potassium to move more quickly out. That's going to be the repolarization. What happens at hyperpolarization is we've had a switch in those ions to such an extent, and those pumps are working so quickly that it overshoots. And then it goes back to resting where you have the leaky potassium gates, but the sodium gates are closed. Once that happens, the cell has been reset and can fire again. These happen very quickly, okay? So the lab you guys did last quarter where you're looking at the neuron of the what was it, the C. elegans or something like that, or nematodes or whatever it was, you can see how the reactions occur very, very quickly. So that is in kind of summary of what is happening with the neuron. So we're going to do a worksheet. The back page is the model, okay? And then all the questions are from
Thank you. Um.
No.
So the fact that the
What did you get for question 3?
I'm just reading it.
I'm still trying to find some way to explain it.
Let me go check.
It's moving down the neuron. look at a neuron. Panel 1 is up here.
And then it's showing the
Well, when it disappears, it comes out. Yeah.
But then like the other ones.
It's the receive.
Yeah. There's nothing on
Yeah, because what is signal? It's A.
I just
It's
think
being
circle.
received at A. The signal is received.
So what's
I poked you in yarn, okay? Where did I poke you? In yarn. That signal is received in your brain. So the brain is there at the end. A is at the arm.
Oh, so it's like the receptor is the response.
Correct. Transduction is B through D or whatever it is. It's transduction. Don't confuse me.
Oh, yeah. Yeah. So it's a, it's like, it's like an, oh, wait, what? So what?
I had it in the bell body text, that's why I was trying to.
Go from positive to negative.
Yeah, so yeah, so it's probably from around it, right?
Direction to sodium ions move through the voltage gated embedded protein. Down. What does the movement of sodium ions across the membrane do to the membrane potential near the open embedded protein?
Yeah, you did. And I was like, no problem, just put it. And I was like, yeah. They put it as a field trip, and I was like, so kind of a smile though.
Oh, one, okay. Oh, gate A is, yeah, so. I just realized, yeah. So, chapter 3, specific gate,
sodium, that's part of
If we go from minus 61 to paragraph 5. Oh, good.
Yeah. Sodium, right, yes, yeah.
Inside of the cell, right? No, right, because it's going from negative to positive.
Only potassium gets pumped away.
Negative.
It's very important.
So just for this case, this one,
Because right here, yeah, you have a higher concentration over there. So at some point, I was going to start using that. The concentration once there's alertness can go down. Like already points out. Remember the question that was from? Yeah, the actual concentration is higher than the size of the
goes against. Sodium,
sodium goes down it.
Putting it is at 30, and then the taxing out of 6. And then you get more out.
Wait for which one? How does that make sense? What hours?
Did you say, 5? Did you say 5? But compared to what?
Oh, wait,
It goes from 80 to 70, yeah, they take
4, 5.
Fully open.
Yeah. It goes negative 80. And it goes plus 35 here, and then it goes on negative 80.
Plus 30. Minus 55. Plus 35. Plus 30. Plus 30. Minus 55. Sodium ion gates closed.
Like, I mean, I see what you're doing.
Yeah. That's what
So,
So then you decrease.
yeah.
I just love sodium. Active transport Wait, no, not trying to put things back. Trying to put sodium back on the outside and pass it on the inside.
Potassium is closed at negative 6.
I know, I was like, what?
Okay.
So they take it back
The last question was really like, is it alive?
A nerve impulse is said to move along the length of the cell. In one or more complete sentences, describe how an electrical signal in the neuron moves.
They showed me the sodium goes up.
when the sodium goes up, then that's the down. That's what
Sodium is the
I feel like I hate writing, because like after I do my grade, as soon as I try to write in actual sentences,
Like my friends like do notes going on.
Oh, he didn't have it. I mean, nothing. Oh, well, I kind of, I think my was wrong. Hold on.
That's a bad
So they remain on the side. So implementing a sodium potassium.
You guys, I remember the photo. I can see images. I don't know what I did.
I said that it would be unbalanced, but it would be closer.
Plus 35 for the.
Oh, yeah, when he's with.
I thought it was like, oh, yeah. Like, it's already chewing on the bottom, now it's on the top, so when the gate's open, the flow's useless. That's just really,
But I get it, I'm maybe like.
I'm not saying that's free, but it's kind of. Oh, yeah, so it's closed at 30.
There's 39 right here. Oh, yeah.
I feel like you bring this up like every corner. You're like, this guy.
Well, no, you said when
Sodium closes and then potassium. And there's also 35. You should just put a bunch of peas in it. Or, cake.
I've never been to Trader Joe's in years. I don't know how sugar I hated it because like it always smells weird.
So,
Yeah, oh, because it's like Costco. Oh. Yeah. I don't know. It doesn't smell like anything. No, the May 3rd is a Saturday. Maybe it's the same.
Oh, wait,
Yeah.
like,
May 3rd is the same.
yeah, it's
Saturday. Okay. Oh, that's good. That means you can't park here without a ticket.
No.
You can't park here?
You cannot park here without a ticket. The school is open on Saturdays.
Why?
No.
They will give tickets.
People sign up for Saturday classes?
Yeah, we run in biology Saturday classes all day.
Why do you?
I didn't know that. I thought it was like
Actually, it's probably most don't give
No,
students
because they didn't
programs.
realize that. Why did you come on Saturday?
Because they're not youth. They're not a they're not young.
But like, I don't even do anything on Saturdays.
They're adults with children and work and night classes.
Right. I forgot about this.
Yeah, they're not children. I'm not trying to be rude, but you're a child.
No.
Angela, it's not you.
Don't worry. I used to it.
They're not
People like Jane, like Mom.
not ambitious young children like us.
Hey, I want a dog. It was already approved.
I always
No.
I only remember because my little brother's birthday is the day after.
Oh, yeah.
Did you have a mechanical pencil? I need
I
I need how to say?
leads.
Leads. Do you have?
I have mechanical pencils, but they're these kinds.
Oh, you don't have a lead 0.7 by any
I
chance?
think I do have some lead actually.
I don't know. I forgot my little bag. And I have 3 pencils. I always carry. Thank you so much. Oh, I get 2.
Take as many as you need. I have like 3 of those.
I'll just get 2. Thank you so much. Appreciate it.
You're always writing, so you'll never know when you run out of leads.
Yeah, I mean, I have a lot. I just didn't bring my little bag, and I always have like 3 pencils, and I only see one.
I just always get these mechanical pencils. And I ditch them every time I run out of leads.
I don't. I like these ones.
That's like around 10.
Or I pay for it, but I just have to think for insurance whenever I pay for it.
I love being a high schooler.
All right, so let's talk about this figure and compare it to this figure, okay? So what's going on? So, threshold, what has happened? Signal has been received, gate opens. Okay? The gate, signal has been received, gate opens. So this is reception, transduction, response. Okay, so response is down there. All right, so signal has been received. What happens? What happens?
Depolarization.
Depolarization. So what moves? The ions. The sodium ions flow in. Okay? At some point, the gates close. What's the voltage for the gates closing? That sounds okay. No, maybe not. Plus 30.
Plus 35.
There we go. Plus 35.
They open
So we close. So we open up, and then the potassium ions are flowing out. And then what happens? It repolarizes. So it's resetting. Okay, so here we have ions are sorted out. Now we have an influx of ions. Ions are starting to move around. Gates are closing, and we start sorting ions out. These are all the channels. What is not shown on this figure? The pumps. The pumps are not shown. Does this process right here, that as shown here, require ATP? No. Depolarization does not require ATP. What requires ATP? Repolarization. The moving of the ions back into the original position. That requires ATP. Okay?
I thought that was decent. Like, where's the model? Where's the model you're talking about? It's also kind of discussion too. I was like,
According to Model 1, what is the membrane potential near sodium ion gates during the refractory period?
It's very low and lost high. It's right next to the sodium peaks and then closed.
Yeah. Because it's, they're clearly moving back.
No, yeah, I don't, because D is whatever, still.
but like, I'm seeing these people after.
Right, so that those are the first, yeah.
Yeah, I thought I
Ricky, can you put back that like action potential picture? Okay, thank you.
So that would be back to the
And it moves
one back.
It's just super low.
They diffuse and go back to different when it gets close. It's like they can't connect. Remember all the sodium ions consume ions are going out. One, go back to that's how all the potassium's already switched out.
I think it's explained, it says how the refractory.
Correct. You wouldn't,
Yeah.
it wouldn't be as low. It would be
So that has to
So what happens if you run Abby?
After because it's one whole between the season or practice, the potential sending is very low.
Yeah, isn't it negative seventy? Yeah. Because when it's negative 70, nothing is flowing.
Isn't it just testing?
Right.
I don't know.
Wait, what's what? What's in
That's what we were talking earlier.
the membrane?
Right.
It's possible to take those ions back out, so the action potential returns or the membrane potential returns.
Carpets. Yeah. That's resting.
Now there was factory.
Yeah, because it opens a negative seven. Or negative. No, they're closed at They close it at. Yeah. Yeah. Oh, this one is a diagram. Yeah.
It goes from 30 to right after the close. That's not very low.
Okay, what question is it?
I've never heard of this teacher.
Yeah, negative 70, negative 81.
And then three
I only have like a mark.
I need to go. I guess we can.
Yes, meet up for pre.
Meet up and mark
It's supposed to be polarization or is it?
Yeah.
You were a rock star?
I think I'm just gonna put it.
There's a missing letter.
I looked it up and I really
I would love to say that I didn't do that.
Too much going on.
I stole like 3 worksheets.
So
You know what? I don't like taking responsibility myself. So I'm like, I'm not sure.
so much hours.
They also switched that to next quarter. Like how your dad was like.
Yes, all I do is I push that into the future. It's a constant future thing.
Have you ever gone to like plant lab?
The plant lab is done. I sent it out.
Oh, right, I forgot about it.
On my computer.
And I thought that's so stupid, I'm so. I said something against them. That was like the worst lab ever shooting. They had negative 70 at the last one. Are you doing it? I do it. That makes sense. Well, I think but I don't know. Wasn't there people in my last one?
Yeah. Jeff teaches that.
Does he? Oh, yeah. Does he go into summer and finish that?
JDE,
I would hate
which would be
that.
this eye. Membrane potential.
We offer bioorganization for some nursing homes.
So we start at negative 70, go up to negative 60, plus 30.
We actually don't think summer's good to do. Like, students do horrible.
It's only good for like the editing classes, I feel.
Negative -70, -70, -60, +30, +35,
Do you know what my favorite thing in the
-80.
world to do is? June 15th, put an away message on my email that says I want it back to September 15th. That's my favorite thing.
They start down September.
I was so happy last year. I know, but that's a student. I'm okay with student. It's the faculty that is out there. Unless they have my phone number, then we can check. But I resigned as the admin last year on June 15th because that was my day out. So wonderful. I went from like 200 emails a day to very few. It was in charge of everything. Yeah. Biology. We just, so Jeff took over the biology, so he's now chair of it. So I resigned from that, which is awesome. And then at STEM, Lev, who starts on May 1st. No, his name's Lev. We hired him.
What is-
We did not hire him.
Well, I was looking at,
Basically, Samita and I have a different buttons for punishment for some reason.
yeah, so it was like, would it be, would you
But
just?
Samita's doing it a lot.
The negative 70 and the negative 60, or is that not right? Yeah, that's what I'm saying.
Because they're university of Washington students, they're like,
Oh, yeah.
that's what I want. Yeah.
Class sometimes maybe take classes and they're like, well, you're doing swim team at the same time, so you're already waking up early for school. And I'm like, I didn't sign up for it.
Mesa?
There's
nothing wrong with Mesa.
Are we supposed to do something?
No. It's resources. You, Marianne, she's nice.
recovers, I guess.
I'm the faculty advisor. Samita and We're both buttons for punishment. She's chair of all the faculty. Yeah, she's faculty president. That's all. I didn't understand why she decided.
I thought she was just like,
Oh no, she's a glutton. Very honestly, the students are the best part of it. Mark just likes to talk.
He comes in and makes fun of us. That's it.
That's also
Oh yeah, we forgot how to use that more.
I should text him that you guys forgot how to do everything. Mark's like one of my favorite viewers because he would like literally pass you out in class.
He scared me. I had him for my first quarter of Gen Chem, and the way he like brings lab notebooks, I was terrified.
He is hilarious. No, he is so funny.
Because he'd come around and like he just stood right in front of me and be like.
I love Mark's when teaching. I love Mark.
When you get Mark and I working together, it's always good. This is like our favorite.
Yeah, that's what I got. A little bit after. But it's like the same.
Here's the thing, you get
Good catch, I know what you said.
Could be a. I really don't know.
Oh, yeah, they start early.
We timed that.
Oh, yeah, take it out.
You're a scientist.
Oh, yeah. I'll be done. August is like prime summer month.
I always hated those because I always like, yeah, friends are so good at
Do you know why? Do you know why it's not reading numbers real? Because they're used to the
Yeah, they keep changing.
and they keep changing.
Yeah, anything in this number.
The pound changing and constantly changing. So it's none of it actually.
If you need it.
There's so much.
One of my friends said because I was like, this makes no sense. I was looking through like all my signs. I was like, there's like 20 of them. And she's like, this is your resting, this is your
Oh,
it doesn't make
every
sense.
day.
Yeah. And she didn't talk to me for a week.
I know.
Oh, yeah.
Yeah, it's just like fun.
Yeah.
Yeah, but like, I don't know, it's kind of like, I find my signs at the same time, I'm like, where is, I remember something like that. I did the same thing.
I don't know before.
Oh, or I said like,
Is it all there? Okay, yeah.
I saw a movie in religion, my mom and grandma's Bible, there was a piece, it's in Bible for her. Yeah, you know, if you don't wanna walk 10 miles, let's just say you will. It is nice for that.
Oh, yeah.
I'm like, no, I do something. You do your
All the stress. They're like in a case. They're just like crazy.
Where's the polarization?
No, yeah. Okay, Vegas. Where is my perfect so dramatic. Yeah, I mean, yeah, they probably need so much money on that. If you're drunk and, you know, you
That's usually what actually
But that's like the high vote. Because hyper polarized, but it's like,
Yeah. Yeah, yeah, that's true. You're
because that's
losing
like
money.
the
Yeah. That's what
I really,
I'm sad. I'm so intrigued by like how they work. Yeah.
get like banned.
They're all Yes. Yes. Yeah, that's literally what I, yeah. Why people be doing that? Like, it's crazy. Yeah, it's in Vegas is like the best place.
Do you guys need a Tesla car?
We probably should.
I'll be there Tesla.
She is.
I know, right? And they were usually pretty good.
What? Yeah, that gives you some downtime by the time it's overpriced.
Yeah, the one in Roseville, they had like near yeah, but now, I don't know.
All right, let's come back together. Got a couple of questions for you guys.
I think it just happens a little bit later.
No, yeah, that's really yeah. No, yeah.
I think I still have to.
Maybe like 10 more seconds. All right, Dr.
I took membrane potential. What do you get?
I did relative refractory period.
Relative refractory period? Okay.
I don't know, it just makes sense.
All right. See about 0.02 seconds apart. This is because there is a relative
Refractory
refractory
period.
period. All right, so if we look at this, what happens? Signals received, sodium, the gate opens, sodium influxes. Sodium continues to influx, and that changes the next voltage gate. That changes the next voltage gate. It's a cascade down. Well, as it moves down, these ones are also starting to experience new things. So the gates are changing, they're moving in a direction. It's going to start resetting. Gates close, and the molecules go back to their original position. So they'll go to position here before they make it to there. Okay, so if you have an action potential that's generated here, it's going to run down this direction by opening up the gates. After it's open, the amount of time it takes to close and reset. Okay? So it is relatively quick, but it still takes time. 10 more seconds.
Oops, what?
There's a question. Okay.
All right? Talk to you. What direction does it move?
Towards the right.
This is the cell body. Okay, this is where we're going to have the nucleus. We're going to have rough ER. Rough ER is going to be manufacturing a lot of the neurotransmitters. Neurotransmitters run down the axon. This is all one cell. Okay? Runs down the axon and gets released here. You receive a signal here, it transmits in that direction to release a signal there, which will be uptake by something else. Okay, which will be uptake by something else. And you'll continue to move down the axons. So our dendrites are going to receive a signal. This could be a chemical signal. It could be an electrochemical signal. It could be a mechanical signal. It could be so many different things. Photons, light, it could be whatever. It's going to receive a signal. It's going to convert said signal into electrochemical signal through a depolarization. Okay? So, you get punched really hard in the arm. Okay? You feel that because something mechanically changed those gates. Those gates now open. A mechanical receptor has been engaged. A chemoreceptor, a chemical is received, opens up the gates. Light receptor, a light is received. Okay? So anything can be received. It's received at the dendrites, is processed and transduced down the axon. Okay? Then you release neurotransmitters at the end, which will then be reuptake by something else. Okay? Could be uptake by a muscle cell. The neurotransmitter in that case would be calcium. It could be, uptake in your brain, dopamine. Right? It could be any number of different things. An axon is a tube. That's all it is. It's this tube. And it's going to have all of these gates here. If we do not encase it in another membrane, what will happen to all of those potassium and sodium ions? They'll go away. Okay? And you'll lose the signal. The signal will disappear. So if I have something where I don't have a membrane right there, I will lose the signal right there. But if I make it really tight, that membrane so tight against it, what happens if one ion moves? It changes everything. Because just like in the mitochondria where I wrap it really tight in a membrane, that's going to insulate it. That's called myelination. Myelination is we're going to insulate that axon. So just like you do with your electrical current, okay? If you have a house that has really good wiring, means the wiring is insulated. It's not going to lose any of those electrons. Just like that. It's not going to lose any of that potential. So I put a probe. I stick a probe here, and a probe here. I engage the signal at this location. Which one is going to detect the depolarization first? This one. Then we're going to detect it here. Then we're going to detect it here. It only moves in one direction. Okay? That allows for signal conduction to only ever be in one direction. Now let's say I initiate somehow in the middle. What happens?
Still.
It'll go in both directions.
Oh.
But you'll only release the neurotransmitter at this end. And then the refractory period will take back over, and you'll probably reset, allowing you to go in one direction. Okay? What is happening here? Depolarization. The membrane is going to become negative.
Mm-hmm.
That is going to allow the sodium to influx. That's going to depolarize here. Then it's going to repolarize. But as it's doing it, these gates are opening, and they're opening as they move down. So the next one will get triggered, then the next one, then the next one. And that really allows it to move in one direction. And that is really key to action potentials. Not all generations will trigger an action potential. Okay? You don't want to be thinking about wearing your underwear. Okay? But if you need to know about your underwear, you need to know about your underwear. I know it sounds stupid, but it's good. So, your underwear moves a little bit, doesn't trigger anything. You're sitting there in your chair, moves a little bit, still not triggering anything. But all of a sudden, you feel something in your underwear. And what do? You freak out because it's initiated a signal. Oh crap, there's something there, and you think about it. Okay? That is what is going on here. Now, some neurons have very low thresholds. Anything is received, it will generate a trigger. So, for instance, if there's the teensiest bit of hydrogen sulfide in this room, every single person's olfactory nerve is going to fire. Because that is an important signal. We need to know that there's H2S in the room. That is really, really bad. But, do I need to know about my underwear? No, so I can lower the threshold. I can reduce it down. So these are your postsynaptic signals. Okay? First one doesn't do nothing. Second one doesn't do nothing. But if I got 20 all of a sudden at the same time, we generate an action potential. Sort of, right? So in your ear, you receive an amplitude and a frequency, so there's a little bit difference. But this is literally like sensory, okay? Something is touching your skin, okay? One thing touching your skin probably is not going to generate it, but if you got a whole bunch of something, now it's going to generate it. Yeah. Your clothes versus feeling a bug, right? Myelination allows this signal to be conducted quickly. So just as we were talking about it, this is a tube, and I have marbles filling this tube. I put one in, one instantly pops out. If I have a hole right here, what happens if I put one in? It's going to pop out in the wrong direction. We're not going to conduct it down. So demyelination is what? Top shirt. What is demyelination? What is a consequence of demyelination? What is demyelination? What is deacetylation?
Loss or
What
destruction.
is demyelination? Removes myelination. All right, next question. We got a couple of questions. Okay, talk to Sharon.
Is the bottom.
D.
Myelination do not have voltage-gated sodium channels and thereby preventing maintenance of the oxygen potentials that propagates down the axon.
All right, if I demyelinate it, means I've removed some of the myelination. Can I still generate action potentials? Yes, so D is factually incorrect. A bare axon would conduct the signal too rapidly. It would cause the signal to dissipate. So C is factually correct. As myelination is lost, there's less ion leakage through the axon membrane. No, there's more leakage. So that is factually incorrect. A is correct. My favorite part is 6 people chose the blank one.
That's because none of them made sense. Can you explain A?
Yeah. So sites of myelination do not have voltage-gated channels. Okay, so that is factually correct.
Okay, I missed that.
Okay, because if I put a marble in here, one immediately pops out if this is full of marbles. So I don't need to open up any more gates for marbles. But as if you have myelination, but now if I don't have myelination, those marbles are going to fall out. So even if I put more in, they're still going to fall out. So if you have a demyelination disease, which nerves are going to be affected? Talk to Joe. Which ones are going to be affected? It's your motor neurons, but which ones first? The longer ones. The ones further away. So you're going to lose the ability to feel your feet first. Then you're going to lose the ability to walk. Then you're going to lose the ability to move your arms. While your brain is still fully functioning because your brain does not have any myelination. Because your neurons in your brain are tiny. The neurons to your toe are huge. So any disruption in the myelination in that 2-meter long nerve will cause a consequence. Whereas this is tiny, not going to have a consequence. Okay? So, if you have a demyelination disease like MS, what happens? You lose the ability to move. Slowly. Yeah, you lose the ability to move slowly, lose the ability to talk, to be able to perceive, express yourself, but you're still able to hear, you're still able to see, you're still able to think. Parkinson's is a little bit different. It's degradation of neurons. Neurons literally are dying. So dementia and Parkinson's, which is really interesting. Dementia is linked to a bacteria in your gut. Parkinson's is also linked to a buildup of certain amino acids, which causes neurons to die. So there's a brand new clinical trial that just is completing in Parkinson's, where they use an enzyme to remove certain amino acids, and it seems to have like 100% efficacy in Parkinson's, which is like fabulous. Dementia is also linked to gut microbacterium. So if you can find a vaccine for that gut microbacterium, you might be able to cure dementia as well.
Can we do that in 6 weeks?
Would be the Nobel Prize. That would be nice. A couple things. Time out. Tomorrow, I will be on Zoom at 10 in the morning and like 12:30 in the afternoon, somewhere in there. I'll post the exact time. That's to talk about cures. You can just do drop-ins. We can talk. Let me know if you're going to show up or whatever. I'll only be on there for like a half hour. Friday, Bio Club. We are leaving here at 11, being at the zoo at 11:30. Bring a lunch. It's supposed to be decent weather. I just looked it up.
Oh, sorry.
get there, we get there. I gotta go see the cracks. Bring lunch. We did not get funding for lunches.
Wow.